Within the intricate tapestry of our DNA, even the tiniest alterations can have profound effects. Microdeletions and microduplications, as their names suggest, involve the deletion or duplication of small segments of DNA, respectively. These changes, while seemingly insignificant in scale, can disrupt the delicate balance of our genetic code, leading to a range of developmental and health issues. Imagine a complex machine with each component playing a vital role. Now picture removing or duplicating a small, seemingly insignificant part. The consequences could be negligible or they could be catastrophic, depending on the part's function. This is the essence of microdeletions and microduplications. Small genetic alterations with potentially significant consequences. These changes occur within our chromosomes, the thread-like structures that house our genes. Genes, in turn, are the blueprints for proteins, the workhorses of our cells. When a microdeletion or microduplication occurs, the number of copies of certain genes is altered, disrupting the delicate balance of protein production. This disruption can manifest in a variety of ways, from subtle learning difficulties to severe physical disabilities. Understanding these genetic alterations is crucial for diagnosing, treating, and ultimately finding cures for a range of genetic disorders. The intricate process of cell division, while generally remarkably accurate, is not immune to errors. Microdeletions and microduplications often arise during meiosis, the specialized cell division that produces sperm and egg cells. During meiosis, chromosomes, each inherited from one parent, pair up and exchange genetic material in a process called recombination. This exchange is a delicate dance, reliant on precise alignment and breakage of DNA strands. Occasionally, however, misalignments occur. Imagine two strands of DNA meant to align perfectly, instead overlapping slightly during recombination. If a breakage occurs in this overlapping region, it can result in one chromosome with a deletion and the other with a duplication of the overlapping segment. This is just one way these genetic alterations can arise. Errors during DNA replication, exposure to certain environmental factors, and even the inherent instability of specific DNA sequences can also contribute to the formation of microdeletions and microduplications. Understanding these mechanisms is crucial for researchers working to develop strategies to prevent or correct these errors. As we delve deeper into the intricacies of our genome, we gain valuable insights into the causes and potential solutions for a range of genetic disorders. The human genome is a symphony of genes, each playing a critical role in our development and well-being. When a microdeletion occurs, it's akin to removing a few notes from this intricate score, potentially disrupting the entire melody. Depending on the genes affected, microdeletions can lead to a range of disorders, collectively known as microdeletion syndromes. One well-known example is Williams syndrome, caused by a microdeletion on chromosome 7. This deletion typically affects around 26 genes, leading to a characteristic set of physical and developmental features. Individuals with Williams syndrome often have distinctive facial features, heart defects, and learning disabilities. They are also known for their outgoing personalities and a love for music. Another example is cri du chat syndrome, French for cry of the cat, named for the high-pitched cat-like cry often exhibited by affected infants. This syndrome is caused by a microdeletion on chromosome 5 and is characterized by developmental delays, intellectual disabilities, and distinctive facial features. These are just two examples of the many syndromes associated with microdeletions. Each syndrome presents a unique set of challenges and requires a tailored approach to care and support. Just as microdeletions can disrupt the genetic symphony, microduplications can also lead to significant impacts. Microduplications involve the duplication of small segments of DNA, which can result in a variety of syndromes. One example is MECP2 duplication syndrome, caused by a duplication on the X chromosome. This duplication can lead to severe intellectual disability, motor dysfunction, and recurrent respiratory infections. Another example is Potocki-Lupski syndrome, caused by a duplication on chromosome 17, which can result in developmental delays, intellectual disabilities, and distinctive facial features. Like microdeletion syndromes, microduplication syndromes present unique challenges and require specialized care and support. De novo mutations are the engine of evolution, driving genetic diversity and the adaptation of species over time. They are the source of new traits, some beneficial, some detrimental, and many with subtle or as yet unknown effects. By introducing new genetic variations into the gene pool, de novo mutations provide the raw material for natural selection to act upon. Beneficial mutations, though relatively rare, can provide individuals with a survival or reproductive advantage. These advantageous mutations are more likely to be passed on to future generations, gradually increasing in frequency within the population. Over time, this process can lead to significant changes in the genetic makeup of a species, driving adaptation to new environments and challenges. Conversely, detrimental de novo mutations can reduce an individual's fitness, decreasing their chances of survival and reproduction. These harmful mutations are less likely to be passed on and tend to be eliminated from the population over time. The balance between these opposing forces, the introduction of new mutations and the elimination of harmful ones, shapes the genetic landscape of a species. Living with a microdeletion or microduplication syndrome can present unique challenges for individuals and their families. The specific challenges faced depend largely on the genes involved and the severity of the condition. Some individuals may experience mild learning difficulties, while others may face significant physical and intellectual disabilities. Early intervention is crucial for individuals with these syndromes. Speech therapy, occupational therapy, and physical therapy can help address developmental delays and improve quality of life. Educational support tailored to the individual's needs is also essential. Support groups and online communities play a vital role in connecting individuals and families affected by microdeletion and microduplication syndromes. These platforms provide a space to share experiences, exchange information, and find emotional support. Microdeletions and microduplications, despite their small size, highlight the remarkable sensitivity and intricacy of the human genome. These minute alterations serve as a powerful reminder that even the slightest changes to our genetic code can have profound and far-reaching consequences. Understanding the mechanisms that give rise to these changes, the syndromes they cause, and their implications for both individuals and the gene pool is crucial for advancing our knowledge of human genetics and developing effective diagnostic and therapeutic strategies. As we continue to unravel the mysteries of our genome, we gain valuable insights into the delicate balance between our genes and our health.